Okay, so let's start with the pre-test version A uh, in the biology subject. So first I have set the Google timer, and which is yeah. So it's coming. Yeah. So I will set 20 minutes. Okay. So let's start and. There is a question given here, that is retest. Oh yeah, use this diagram to answer the following 6 questions. Okay, let's go. uh, there are several marked letters are there. So first question is that level G. Level G, yes, that is level G. Level G represents several proteins that assist with cell division, protection, and maintenance of cells. What do these proteins comprise? Cell body, cytoskeleton, head, musculoskeleton system. So G, yes, that is G. It represents several proteins that assist with cell division, this protection, maintenance of cell safe. So I think this will be cytoskeleton. Next, level F represents which, and that is necessary for what? Level F, that is, yes, this part. So level F, what is this? Okay, I can guess it. I think the options are centrioles, cell division, centrioles, the quiescence phase, microtubules, quiescence phase. So I think it is centrioles, and which is necessary for cell division. Yes, I think this is the correct answer. Okay, so next is the organelle represented by level D. Level D means that is level D. Okay, so it's mainly found in which portion? And your answers should be the names of the organelles. Separate your answers with comma and then space. Level organelle represented by level. So I think level D, uh, found in cell. I think found that is in cell, and I think the name of the organelle should be that is in between the cell definitely. And so what is the name of it? So I think it will be cytoplasm. I guess it. I am actually not confident about it because I have not in touch with the biology subject for a while. So I think it will be cytoplasm. Cytoplasm. So separate your answers with a comma and then a space. So I think organelle represented by level D is mainly found in cell. I think in cell. Okay. So next question is that the main function of the Golgi apparatus represented by level which is what? Golgi apparatus. Golgi apparatus in this picture, I think Golgi will be yes K. Definitely it will be K. And the question is that yes, K is there in our option. So represented by level K, and is what is its function? Sending and receiving modified proteins. I think it will be the correct. Lipid synthesis or sending and receiving modified protein. I think Golgi apparatus. I think it will be K and sending receiving modified protein. So now the question is the organelle level C is present in level C. Oh my god, what is this? Level C. On level C, what will be what are the options? Organelle level C is present in animal cells only, plant cells, animal and plant cells, prokaryotes only. So organelle level C is present in which cell? Animal cell, plant cell, animal and plant cell, prokaryotes. So this I think uh, I think this will be so I have not idea of it, so I guess this will be in both animal and plant cell. Okay, so organelle labeled ACEH that will be in higher number in muscle cells compared to fat cell will be in higher number in muscle cell compared to fat cell. ACEH, and H. Okay, that is I think lysosome. some fat. E means CAC. Think A. Think A. Next. So just as an amino acid is a monomer of a protein, this is a monomer of DNA. Just as an amino acid is a monomer of a protein and is a monomer of DNA. Nucleotide is a monomer of DNA. Site. I think nucleotide. Pentose. No. A DNA monomer is called nucleotide, and yes, and is made up of three parts: a nitrogen base, deoxyribose sugar, and sulfate. Which part is incorrect? Sulfate group. That definitely. DNA is more stable than RNA. I think DNA is more stable, and therefore DNA is more stable. So than RNA. Therefore, more reactive or less reactive. DNA, deoxyribose. DNA is ribonucleic acid, deoxynucleic acid, ribonucleic acid. Sorry, I think more reactive. Just give your answer in the previous question. DNA is more stable than RNA, and DNA is more stable than RNA, and so it will be more reactive than RNA. Okay, next. Oh, what is this? Use this diagram to, I think, to answer the following 7 questions. According to the strandedness of this molecule, I think the DNA strandedness. What type of nucleic acid is? DNA, RNA, NTP, and NTP. Use this diagram to answer the following 7 questions. Delta codon, C for codon, I think. CGA, the strandedness. CAUG, AUG, C delta. Nucleic acid, DNA, not RNA, DNTP, NTP. Okay, I have not idea about it. So, according to the types of bases in the molecule, what type of nucleic acid? So, I think RNA. What is paradoxical about this image? What is paradoxical? I think it is anticodon. According to your answer to question 11, what bases do delta and psi represent? Delta and epsilon. Okay, I have not much idea about this because I've not studied this codon or anticodons recently, or it has been studied in the high school class, but I'm not in touch with this. So I think it will be 80. According to your answer question, what percentage represent? Green and New Brazil. So use the following diagram along the previous to answer the following two questions. This is a sugar from the previous nucleic acid, and there is a hydroxyl bonded 2 prime 3 5 prime. What type of nucleic acid it is? CH2OH prime. I think it is deoxyribose DNA. What conclusion can be drawn from your find? DNA can sometimes include uracil. The backbone of DNA can sometimes include this type of thing. It does not matter. RNA can sometimes be found double-stranded. So the ordinary flow of information in a cell, separate each answer with a comma then space. Flow of information in a cell. Information. I think tRNA. tRNA. No, sorry. tRNA, comma mRNA, comma, and another is that rRNA. I think this will be the order. The process that takes place at the first arrow is the process second arrow. Transcription, translation. I think transcription and translation. Okay, which statement about proteins is correct and alpha helix is indicative of primary structure, beta sheet is indicative secondary structure. Multiple proteins interacting is indicative tertiary structure. Quaternary structure is only theoretically possible. Okay, so this is actually codon part. DNA template that contains 5 prime, I think 3 prime to 5 prime. mRNA strand choice must reversed. Compared to answer DNA and strNA 5 prime to 3 prime. By defining which protein sequence is meant to code for which protein sequence. What protein sequence does it mean to code for? G L N. Okay, I have not idea about this. I go to next. Point mutation causes an amino acid to change to a stop codon. What is the effect of the resulting polypeptide? No change, truncated non-functional, W goes to nucleus. I think truncated and non-functional. Second point mutation causes that stop codon to change into codon that is different from the original. What is this mutation called? Silent nonsense missense missense. Okay, 12 out of 25. So next part starts and that is about 30 minutes. Okay, I will start it. Uh, 30 minutes. No, this is not. Okay. Okay, next 30 minutes starts and then yes, that is correct. The level G represents several proteins that assist with cell division, that is cytoskeleton, and level A represents that is also correct. Organelles, correct answer. RAF ER, not cell. Okay, or RER. Yes, yes. Reticulum. I think endoplasmic reticulum. That should be uh, rough endoplasmic reticulum and cytoplasm. So only one is correct. So not two is correct. Next, the main function of the Golgi apparatus. Okay, so I can check it. This copy and then okay. So actually the question is the organelle represented by level D mainly organelles. What is, I think we can search what is uh, RER in organelles. Sorry. It is RER in organelles. Organelles. Organelles in cells. What is organelles? Okay, this is rough endoplasmic reticulum and is a cellular organelle characterized by ribosomes on its surface, giving it a rough appearance. So mm, endoplasmic reticulum is a part of transportation system of eukaryotic cell and has many other important functions such as protein folding. The word endoplasm within the cytoplasm, yes, and reticulum is Latin for little net. It is a type of organelle made of two subunits. First one is rough endoplasmic reticulum and smooth endoplasmic reticulum. RER and SCR. The endoplasmic reticulum is found in most eukaryotic cells and forms an interconnected network of flattened membrane and closed sacs known as cisternae. Smooth endoplasmic reticulum, and the endoplasmic reticulum is found in most eukaryotic cells and forms an interconnected network of flattened membrane enclosed sacs known as cisternae and tubular structure. In the SCR, the membrane of ER are continuous with the outer nuclear membrane, and the endoplasmic reticulum is not found in red blood cells or spermatozoa. So actually, endoplasmic reticulum is this two type of ER that share many of the same proteins and engage in certain common activities such as synthesis of certain lipids, cholesterol. Different type of cells contain different ratios of two type of ER depending on the activities of the cell. RER is found mainly toward the nucleus of the cell and ACR towards the cell membrane or plasma membrane of cell. The outer cytosolic phase of the RER is studied in the ribosome that are the sites of protein synthesis. RER is found specifically prominent in cells such as hepatocytes. So it was observed by ER is observed by light microscopy by Charles Garnier. Okay, so this is the actually structure of endoplasmic reticulum. Okay, the surface of rough endoplasmic reticulum, also we can say granular endoplasmic reticulum, is studded with protein manufacturing ribosome, giving it a rough appearance. And the binding site of the ribosome on the rough endoplasmic reticulum is the translocon. That is 7 and 8. Okay, 7 and 8. Yes, that is 7 and 8. Okay. This special complex forms when a free ribosome begins translating the mRNA of the protein. So, mRNA of a protein destined for the secretory pathway. First 5 to 30 amino acid polymerase they encode a signal peptide. So, translocation pauses and the ribosome complex binds to the RER translocon, where translation continues with the nascent protein forming into the RER lumen membrane. Okay, that gives little about idea of what is RER. Next question. Okay, so next is that, yes, that's correct. The main function of Golgi apparatus represented by level is case sending receiving modified protein. Organelle level C is animal plant both cell. Organelle labeled will be in higher number muscle than computer, which will be H. So what is actually H? I think lysosome. So which remain the organelle which remain in muscle cell compared to fat cell. Okay. The organelle, the organelle which remain remain maximum, I think, higher number in muscle cell, which remains in higher number in muscle cell than in fat cell. Mitochondria, okay, that is more in muscle cell than fat cell. What will be the muscle cell require large amount? Okay. Mitochondria, this is mitochondria, so the structure should be this H. Yes, yes, that is mitochondria. Okay, that is mitochondria. So mitochondria is an organelle found in the cells of most eukaryotes, such as animals, plants, and fungi. Mitochondria have double membrane structure and use aerobic respiration to generate adenosine triphosphate, which is used, that is ATP, is used throughout the cell as a source of chemical energy. That is the important point. They were discovered by Albert von Collier in 1857 in the voluntary muscles of insects. The term mitochondrion, meaning thread-like granules, such like that, was coined by Carl Binder in 1898. The mitochondrion is popularly nicknamed the powerhouse of the cell, a phrase popularized by Philips Sienkiewicz in 1957 Scientific American article of the same name. Some cells in some multicellular organisms lack mitochondria, for example, mature mammalian red blood cells. The multicellular animal Heningua salminicola is known to have retained mitochondrion-related organelles despite a complete loss of their mitochondrial genome. A large number of unicellular organisms such as Mitosporidia, parabacilliates, and diplomonads have reduced or transformed their mitochondria into other structures. Okay, mitochondria are commonly between 0.75 and 3 micrometers square in cross section but vary considerably in size and structure. Unless specifically stained, they are not visible. The mitochondria is composed of compartments that carry out specialized functions. These compartment or regions include the outer membrane, intermembrane space, inner membrane, Christie matrix. In addition to supplying cellular energy, mitochondria are involved in other tasks such as signaling, cellular differentiation, cell death, as well as maintaining control of cell cycle, cell growth. Mitochondrial biogenesis is in turn temporarily coordinated with this cellular process. Mitochondria are implicated in human disorders and conditions such as mitochondrial disease, cardiac dysfunction, heart failure, autism. Number of mitochondria in a cell vary widely by organism, tissue, and cell type. Mature red blood cell has no mitochondria, whereas liver cell can have more than 2000. Almost most of the eukaryotic cells' DNA is contained In the cell's nucleus, the mitochondria has its own genome that is called mitogenome, that is similar to bacterial genomes. This finding has led to general acceptance of endosymbiotic hypothesis that free-living prokaryotic ancestors of modern mitochondria permanently fused with eukaryotic cells in the distant past, evolving such that modern animals, plants, fungi, and other eukaryotes respire to generate cellular energy. The structure is the outer mitochondrial membrane, the intermembrane space, the inner mitochondrial membrane, the Christie space, the matrix space. That is the important part. Outer membrane, inner membrane. Okay, okay. So inner membrane uh, function is that perform the electron transport chain redox reaction ATP synthase which generates ATP in the matrix, specific transport protein that regulate metabolite passage into and out the mitochondrial matrix, and the main function is energy conversion. Okay, so next question is that next, yes, this is correct. Just as an amino acid is a monomer of protein and nucleotide is a monomer. DNA monomer, that is also correct. DNA is okay. This is less stable. Stability of DNA and RNA. Okay, I can find it that in this way. The stability of DNA and RNA. DNA is more stable due to its double-stranded structure and presence of deoxyribose sugar, making it suited for long-term genetic. RNA being less stable is suitable for short-term tasks like transferring genetic information from DNA to synthesis. So this is stable due to the double-stranded structure and the presence of sugar, deoxyribose sugar, making it suited for long-term genetic storage and a short-term task performing transferring genetic storage, and this transfer genetic information from DNA to during protein synthesis. So why RNA is less reactive than DNA? Why? What is the reactivity? What is the reactivity of DNA and RNA? RNA is nearly more reactive than DNA due to presence of 2-hydroxyl group on its ribose sugar, making it susceptible to hydrolysis. RNA is more reactive, containing ribose sugar is more than DNA and is not stable in alkaline condition. And RNA's larger helical groups mean it is more easily subject to attack by enzymes. Okay, so main point is that RNA containing an ribose sugar is more reactive than DNA and it is not stable in alkaline condition. So RNA's large helical um, groups mean it is more easily subject to attack by enzyme. RNA is more sensitive because and it is primarily single-stranded and often folds into complex three-dimensional shapes necessary for its function. So this single-stranded nature makes RNA to chemical and more susceptible to chemical and enzymatic attacks. Okay, that is the point. So DNA is more stable but less reactive than RNA. Okay. So this also incorrect. This will be DNA. So using this diagram, so according to the standardness, what type of nucleic acid, that will be DNA. Okay, so DNA stranded strandedness of DNA, and its structure. So 355 nitrogen base adenine, thymine, guanine, cytosine, and these are the base pair, and these are the sugar phosphate backbone. So these are the actually strandedness of DNA. So sugar phosphate backbone and base pair adenine, nitrogen base thymine, guanine with cytosine, thymine with adenine, adenine with thymine, cytosine with guanine, adenine, thymine. Okay, so DNA molecule have an antiparallel structure, that is the two strands of the helix run in opposite direction of one another. So each strand has a 5' end and a 3' end. Solving the structure of DNA was one of the great scientific achievements of the century. Okay. Okay, so I'll go to the next part. So according to the types of bases, this is correct. What is the paradox elements? Double-strandedness is indicative of, yes, that is the double-strandedness. Double-strandedness is indicative of DNA, while the presence of uracil indicates RNA. That is right. Based on these two pieces of information, it is inconclusive or something similar. This is correct. This is also correct. Uh, this is RNA. This sugar from previously discussed nucleic acid. There is a hydroxyl bonded to 2 prime, 3 prime, 5 prime carbon. What type of nucleic acid? So this will be correct answer will be RNA. Oh, this is the structure of nucleic acid. C2H, C2H. DNA and RNA are the two most important molecules in cell biology, but what are the key differences? Yes, you can say nucleobases of DNA. That is guanine, adenine, cytosine, thymine. These are the base pair, and nucleobases of RNA. Guanine, adenine, cytosine, uracil. Both are same in place of thymine, that is uracil in RNA. Okay, and the question is that this is a sugar from previously discussed nucleus. There is a hydroxyl bonded to 2 prime, 3 prime and 5 prime. So okay, there as it is a 2 prime, then 3 prime and 5 prime, so it will be RNA. Okay, that is the correct answer. So next incorrect is what conclusion can be drawn from your finding? Uh, RNA can sometimes be found those. Okay, so RNA can sometimes be double stranded. Yes, while RNA is typically single-stranded, it can indeed form double-stranded structure in certain contexts. These include double-stranded RNA virus, okay, where the viral genome is composed of double-stranded RNA. Additionally, RNA molecules can fold back on themselves, creating double-stranded regions within a single strand through intramolecular base pairing. Okay. The ordinary flow, okay. Ordinary flow of information in a cell is DNA, RNA, then protein. Separate each answer with comma. Okay, that will be DNA, RNA, and protein. That is correct transcription translation. About the correct alpha helix is indicated. Beta alpha helix indicated prime minister. Okay, that is not correct. Okay. No, alpha helix is not indicative of the primary structure of a protein. It is it. Oh, okay. Alpha helix is a secondary structure. The primary structure refers to the sequence of amino acid in polypeptide chain. The alpha helix is a specific three-dimensional conformation formed by the twisting of this chain. So what will be the beta sheet? Beta sheet, beta sheet. No, beta sheet also not a primary structure, it is a secondary structure. Okay, so nah, multiple protein interacting is indicative of structure. Okay, and this is the codon part. Okay, this will be glutamine, threonine, leucine, valine, and leucine. And the point will truncate. Okay, okay, move to next part. That will be another 30 minutes. And there's some questions are there that is given here. Okay, so first 30 minutes completed, then next 30 minutes put up some questions in the Google, that is animal cell diagram. Animal cell diagram. So animal cell diagram typically shows a rounded irregular shape enclosed by a plasma membrane. It contains various organelles like nucleus containing DNA, mitochondria for energy, and other structures like endoplasmic reticulum, ribosomes. Unlike plant cell, animal cells lack a cell wall and chloroplast. So, more detailed look at the key components: plasma membrane and the outer boundary that regulates the movement of substances in and out of the cell, nucleus, the control Center containing the cell's genetic material DNA, cytoplasm, the gel-like substance within the cell where most of the cell's activities takes place, mitochondria, the powerhouse of the cell responsible for producing energy, and other organelles such as endoplasmic reticulum involved in protein lipid synthesis, and ribosome for protein production, Golgi apparatus modifying and packaging proteins, lysosomes containing digestive enzyme, and centrioles involved in cell division. So these are the structures of animal cell structure. That is nucleus, that is area, that is endoplasmic, rough endoplasmic reticulum, and this is smooth part, smooth endoplasmic reticulum. This is centrioles, that is Golgi apparatus, and that is the ribosome, power, and then this is the cell membrane. This is the cytoplasm, and this is a vacuole, microtubules, peroxisome, mitochondria, lysosome. Okay, let's get in touch to the next question. That is plant cell diagram. Okay, let's check it. Uh, Plant cell diagram with uh, plant cell diagram typically includes structures like cell wall, cell membrane, nucleus, chloroplast, mitochondria, endoplasmic reticulum, Golgi apparatus, ribosome, vacuole, cytoplasm. These components work together to carry out essential functions such as photosynthesis, energy production, and storage. So, yes, this is the plant cell. We can see this is the cytoplasm, this is lysosome, and this is the mitochondria, powerhouse of energy, vacuole, peroxisome, and vesicle, Golgi apparatus, cell membrane, plasma, cell wall, nuclear envelope, nuclear pore, nucleolus, nucleoplasm, smooth and rough endoplasmic reticulum, ribosome, chloroplast. So actually the difference, what is the basic difference between plant and animal cell? What is the basic difference between plant and animal cells? Plant and animal cells are both eukaryotic, okay, but they have key structural difference. Plant cell possesses a rigid cell wall, okay. The cell wall is rigid in case of plant cell, chloroplast, and large central vacuole, while an animal cells lack this feature, okay. Here the in case of animal cell, the cell wall are more soft, and central vacuole is little smaller, and instead animal cells have smaller, more numerous vacuole centers which are involved in cell division. Okay, these are the basic difference. Thanks. Next question is that functions and function of cell organelle. Function of cell organelles. So, what are the 10 functions of cell organelles? 13 organelles and their function. Nucleus that stores genetic information. Stores the genetic information. Organelle function type and importance. So, yes, there are many different organelles in different cell. The organelle function charts given that is very beneficial for us. That is such as some nucleus. Its cell type is eukaryotic and function stores genetic information. Nucleolus, that cell is also eukaryotic, makes ribosome. Cytoplasm, all cells in there contains the content of the cell. Very good information from this. We get the substantive information from there. Cytosol, that remains all cells. It functions gel like matrix that holds water and nutrients. Cytoskeleton, that is remains in eukaryotic and it acts to determine the structure, support and transport. Ribosome in all cells makes protein. RER eukaryotic makes protein for endomembrane system. Smooth ER eukaryotic cell detoxifies the cell and makes lipids. Golgi apparatus eukaryotic cell sorts and sieves protein. Mitochondria makes energy. Lysosome eukaryotic animal cells only removes unwanted material and waste. Peroxisome eukaryotic regulate biochemical pathways that involve oxidation. Vacuoles eukaryotic store water and nutrient. Vesicles eukaryotic transport materials around the cell. Cell membrane all remains in there and a thin flexible barrier that separates the cell from its environment. Cell wall, plants, fungi, prokaryotes, rigid barrier and that protects the cell. Large central vacuole, plants only, stores water and regulates target pressure. Chloroplast, plants only, makes food using the process of photosynthesis. Okay, so this is good point. Next is what is the difference between RNA and DNA? RNA, DNA differ in their sugar molecule. Deoxyribose in DNA, ribose in RNA, nitrogenous base thymine in DNA and replaced by uracil in RNA. DNA is typically double strand while RNA is single strand. These are the basic differences. Deoxyribose, sugar and ribose. So this is close type of question. Okay, so we will check it. Deoxyribose and ribose sugar. Deoxyribose and ribose both are 5-carbon sugar pentose found in nucleic acid, but they differ in the presence of hydroxyl group. That is the important point on the 2' carbon. Ribose has the wedge group while deoxyribose has a hydrogen atom instead. Yes, that is the important point. Ribose has the wedge group while deoxyribose has the hydrogen atom instead, hence the name deoxy, meaning lacking oxygen. This seemingly small difference has significant implications for the structure and function of DNA and RNA. So we can see this picture that in deoxyribose, that is, both the structure are same, but we can see 1 2, this is 2 prime end. In case of DNA, 2 prime end, that is a hydrogen, and in case of ribose, 1 2 2 prime end, there is a OH group. This is the basic difference. So this molecular formula DNA C5H10O4, and here it is C5H10O5. One excess oxygen is there in case of RNA. Very good question and very good answers are given. Yes, that is one of the important central dogma of biology. Central dogma molecular biology describes the flow of genetic information within a biological system, generally from DNA to RNA protein. This means that DNA serves as a template for RNA synthesis, that is transcription, and RNA then directs protein synthesis, translation. While this is the common pathway, exceptions like reverse transcription exist, particularly in some viruses. DNA to RNA, that is called the transcription, RNA to DNA, that is translation. Actually, it sends the genetic information to RNA. Okay, that is called the transcription, and RNA sends the proteins, directs the proteins, and that is called the translation. Okay, good question. Next is that, okay, next is to answer all these questions. Okay, let's check it. So these are the practice questions. What part of the cell contains the instruction for life? Uh, Golgi apparatus, cell wall, nucleus, nucleolus, lysosome. What part of the cell contains the instruction for life? Instruction. So I think it will be nucleus. Then the smooth and rough endoplasmic reticulum both synthesize a molecule. Smooth produces protein synthesis, I think. Lipids. Lipids and produce a protein. Lipids, proteins. So smooth produces lipids and the rough produces proteins. Which organelle is necessary for cell division? For cell division, mm, okay, I think it will be cytoplasm. Which organ is necessary for cell division? It will be cytoplasm. Choose a symbol on the keyboard that best corresponds to DNA. Choose a symbol on the keyboard that best corresponds to DNA. Hmm. DNA's gene. So, what should be symbol? Symbol. Symbol means yes, that should be given. Choose a symbol on the keyboard that corresponds to RNA. Okay, the symbol. Okay, that should be in case of RNA. Okay, let's move it. I think the symbol will be hmm, helical pattern. Central dogma biology is synonym for the flow of information in a cell. What is it? DNA to RNA. DNA describes flow of genetic information in a biology DNA to RNA to protein. That is the correct answer. In eukaryote DNA is scattered throughout the cell. DNA is scattered. False. In DNA, A pairs with thymine. What is the effect of a mutation that does not change the amino acid that the codon codes? What is the effect of a mutation that does not change the amino acid that codon codes for? No failure and no and protein produced or organism. Nothing. The two main type of secondary structure are alpha and beta are seeds, alpha helices and seeds. Let's go. 7 out of 10. Yes, that is correct. Centrosome and centriole. Okay. Which organ is necessary for centrosome and centriole? Symbol is equal to. Okay. So there's so here the which organelles is necessary for cell division and which organelle is necessary for cell division. Okay. In animal cell, the centriole and more broadly the centrosome is the organelle essential for cell division. Centrioles are cylindrical structure that organize microtubules which are critical for forming the spindle fibers that separate chromosomes during mitosis. While plant cells lack centrioles, they achieve more function through other structures like the cell plate. So why centrioles are important? Because spindle formation. Centrioles duplicate before cell division and migrate to opposite poles of the cell, forming the spindle apparatus. Chromosome segregation. The spindle fibers formed from the microtubules organized by the centrioles attach to the chromosome and pull them apart, ensuring each daughter cell receives a complete set of chromosomes. That, that is important point. Animal cell specific. While some other organisms can divide without centrioles, they are crucial for the process in most animal cells. And in case of plant cells achieve a similar result through the formation of a cell plate, a structure that divides the cell into two. Okay, another wrong answer is that uh, what is the symbol on keyboard? What defines equal to? what defines equal to in DNA? Symbol defines DNA. Not definition of DNA. So DNA is molecular in the keyboard. DNA does not have chemical symbol like element. It is written by name. DNA stands for is complex as self-genetic blueprint living organism. Okay, that's it. So we will move to the next section. The next part is that this 15 minutes almost done. So next part is that post. Okay, we'll move to the next part. Next part is 20 minutes. Okay, now start it. So next part is that post test version B. So version B is given here. Wait just a minute. Yes, that is the version B. I think this is okay. Oh, cell structure. So use this diagram to answer the following 6 questions. Let's take it. Let's start it. So there are different labeled parts. Level 6 is this, represents an organelle that produces sugar molecule. What is it called? Sugar. Level 6 represents an organelle that produces sugar molecule. Sugar molecule. Okay. What is this? Mitochondria, Golgi bodies, sugar molecule. Sugar molecule means uh, Golgi bodies, chloroplast, vacuole. Mm, so, according to our knowledge, we know that the Golgi apparatus sorts and sieves proteins, and chloroplast makes food using the process of photosynthesis. So question is that it produces sugar, mitochondria, Golgi bodies, chloroplast, vacuole. Mitochondria, it makes energy. Chloroplast, vacuole store water and nutrient. proteins. Chloroplast means makes food using the process of photon. Produces. Okay, I will go for mitochondria. Next, level 4 represents level 4. 4 means this. Yes, this is the which is where I think this is vacuole. Vacuole for holding nutrient monetarity physical. I think it will be vacuole holding nutrient. So 4 means this is the 4 central vacuole or vesicle transport material and vacuole store water. So vacuole and vesicle. Vacuole store water and nutrient, and vesicle actually basically transport material. So I think this will be vacuole holding nutrient. Level 10 is the internal layer of the substance that surrounds the cell. This is called the cell. Level 10. 10 means 10, 10, 10. Cell wall, I think. Cell wall. The main function of the smooth endoplasmic reticulum represented by level smooth that actually protein lipid synthesis I think smooth lipid synthesis. So here the option is 219. Yes, 9 is different. 2 is not smooth. 9 is smooth, so this will be 9. 9, that is lipid synthesis. Level 5 present in level 5. 5, 5, 5. Animals have some progress, some precursor. Animal cell. The organelle labeled in 15810 will be higher number in muscle cell. That is mitochondria. 15810. 158. 15810. No, that will be 1. Okay. The backbone of a polymer of nucleotide contains ribose sugar. Backbone of a polymer of nucleotide contains ribose. What is this polymer? That is RNA. A DNA monomer is called nucleate and is made of 3 parts. Which part is correct? Which part is correct? Nitrogen-based ribose sugar. Ribose sugar. Which part is correct? Nitrogenous base. This will be deoxyribose sugar and phosphate group. Why is RNA not the main carrier of genetic material? RNA single, so it carries half of the information. RNA is less stable. RNA is less stable than DNA. Uracil is toxic and RNA cannot belong. Genetic material cannot less stable, or RNA is single stranded. That is the correct that RNA is single, so it carries half the information. It actually transfers the protein information. So RNA is less stable. I think stable. DNA is more stable than RNA, but less reactive than RNA. As RNA is less reactive, so it will form complex structure. According to the standard of what type of nucleic acid? DNA. According to types of bases of the molecule, what type of nucleic acid? Oh no, this will be RNA. According to the types of bases in the molecule, কি base রয়েছে? Thymine. So both will be. Thymine should be DNA. What is the paradox about this image? This is single stranded. This is single stranded. According to the answer to question, what must be the final part of the molecule? Phosphate, nitrogen base, and ribose sugar. According to your answer, what is the final part of the phosphate, nitrogen base, and প্রথমে কি দিলাম RNA and then DNA, deoxyribose sugar. Use the following information along with the previous to answer the following two. After testing, this nucleic acid is found to be the entirely of thriving as a full allen genome. Chargaff's rule still apply. Deoxyribose sugar is found in its background. After testing, this nucleic acid is found to be the entirety of a thriving and successful alliance genome. Chargaff rule still apply. Deoxyribose sugar is found in its backbone. Delta is and AT respectively. What conclusion can be drawn from here? A genome that is compressed single-stranded DNA is a sufficient as double. Genome needs CG to reproduce. This type of sugar does not matter. This must be exception of Chargaff. Reverse transcription is the inverse process of ordinary central dogma of biology. It can be represented as DNA to RNA. The inverse of the process described in the previous question is reverse transcription. Straight chain of amino acids indicative primary structure. Beta spiral is indicative of secondary structure. Protein bonding with another indicative tertiary structure. Quaternary structure completely. Okay. Oh. So DNA template strand contains sequence 3' GTA, TTG, GCT, GTG, 5'. What is the complex strand of mRNA? What is the most likely reason that the sequence in the question has been written in reverse order of the it is not going the template strand is 3' to 5', but the mRNA strand is what protein sequence does this amino code for? Okay. Let's find the possible protein sequence that will be CAU, 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 HIS, histidine. Okay, next AAC, 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 AAC. So AAC, ASN, ASN. Next, U, triple U. Triple U is PHE. PHE. Next, CGA. 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 CGA is not there. CGA arginine. CSC. CSC. Histidine CSC. Okay, time completed. Hmm. Okay, now move to next. So point mutation has occurred but the polypeptide is same. What is the mutation called? Point mutation has occurred but the polypeptide is same. Silent. Point mutation occurs separate for the second one instead first one causes original what type of mutation? Missense. Okay, 16 out of 25. So last part is that so here it should be chloroplast. Okay. Which is central vacuole maintaining turgidity. Okay. Level 10 is membrane. Some eukaryotes. Level 5 present in some prokaryotes and some eukaryotes. More. One more hydroxyl than DNA, so it is less stable. RNA, DNA. This single stranded nature indicates as RNA, while the type of bases indicates RNA. It is quite correct, this actually. Straight genome is primary structure. Yes, that is also correct. Silent, that is missense. Nonsense. Okay, so last part is that quick survey systems usability scale. Which model did you sense? Did not use model. Next, how mentally demanding was this task? Uh, Very high demand. Okay, I will give 9. How physically demanding was this? Not physically demanding. So this is some physical atmosphere is equal, so I will give 3. How hurried or rushed was the pace of this task? Medium, 50. How successful were you in completing your task? I will say about 9. How hard did you have to work to what you did? Right. How discourage digital stress and add value? Very little. Okay, thank you. So this actually ended here or completes this survey. Okay, thank you.